10,000 papers. That's what shows up when you search your topic. And everyone says, just read more. Terrible advice, impossible advice. Here's what actually works: a system to find exactly 15 anchor papers in 1 hour. These 15 papers, they carry your entire literature review. Today I'm showing you how to cut 90% of your screening time while keeping your data completely private. No spreadsheet nightmares, no drowning in tabs. Let's fix this. Picture this: you type your topic into a database. What happens? You get buried. 10,000 results, sometimes more. And here's what nobody tells you: that mountain of papers isn't helping, it's paralyzing you. The average researcher spends 52% of their time just searching and organizing. Not thinking, searching. More than half your brain power wasted on sifting through noise. The problem isn't finding information, it's drowning in it. Your job isn't to read everything, it's to find what matters. Here's what's frustrating. You'd think more searching makes you smarter, right? More papers, more knowledge. Nope. Every new search adds another brick to the wall. You're not learning faster. You're hitting a barrier faster. I've watched brilliant people spend entire weekends clicking through abstracts, opening tabs, losing tabs, forgetting what they already read. End of the weekend? More confused than when they started? The search engines aren't broken. The approach is broken. You need a filter, not a fire hose. Here's the shift. You don't need 10,000 papers. You need 15. That's it. 15 anchor papers that define your entire field. These are the studies everyone cites, the ones that shaped how people think about your topic. How do you find them? Look for patterns. Papers that show up again and again in reference lists. Once you nail those 15, you've got the foundation. Everything else is building on them or repeating them. Stop chasing every paper. Start finding the ones that actually matter. This sounds impossible. It's not. You can find those 15 anchor papers in 60 minutes. 1 hour. I know, you've been drowning for weeks, but it works if you have a system. The key is ruthless filtering. You're not reading anything yet. You're scanning titles, checking citation counts, looking at dates. Think of it like speed dating for research. One question: is this paper foundational or forgettable? Most are forgettable. That's fine. You're hunting for the 15 that carry your entire literature review. Listen, I need to be honest. The old way, reading everything manually, tracking it in spreadsheets, that approach drains you. Researchers burn out not from thinking, but from organizing. Copying titles into cells, color-coding rows, losing track of which papers you already rejected. It's not just inefficient, it's exhausting in a way that makes you hate your own research. And that's the real cost nobody talks about. Your enthusiasm dies in Excel. We're done with that. Check this out. Speed requires a system, what I call the research stack. 3 tools working together. First, the database. Search across PubMed, Scopus, Web of Science all at once. Second, the sieve. Removes duplicates and junk automatically. Third, the mapper. Shows you how papers connect to each other. When these 3 work together, you stop thinking like a human and start thinking like a machine. Not cold, efficient. The stack does the boring work so your brain can do the interesting work. That's the whole point. Real talk about privacy. Every time you upload research to some cloud AI tool, you're handing over your ideas, your notes, your half-formed theories. That data goes somewhere, gets stored somewhere, trains someone else's model. It's not paranoid to worry about this. It's smart. What you need is a system that runs locally. Your data stays yours. No uploads, no mystery servers. This matters if you're working on something original. Protect your work before it becomes someone else's training data. Meet Feynman, named after the physicist who said if you can't explain something simply, you don't understand it. It's your private desktop tool for research, runs entirely on your machine. Nothing leaves your laptop. It handles the database, the sieve, and the mapper all in one place. Think of it as a research assistant who never sleeps, never forgets, and never shares your secrets. Your literature review goes from nightmare to something you can actually finish. Love it. Phase 0, the great deduplication. Before you do anything smart, do something simple. Delete the clones. When you search PubMed, Scopus, and Web of Science for the same topic, massive overlap. Same paper appears 3, 4, 5 times with slightly different metadata. Feynman catches all of that automatically. merges duplicates, spits out one clean master list. No more wasting time realizing you already rejected a paper yesterday under a different title. This step alone saves hours of confusion. Stage 1, the 3-button blitz. High-speed triage. You're looking at abstracts with 3 choices: yes, no, or maybe. That's it. No deep reading, no agonizing. Gut decisions based on whether the paper fits your criteria. Green for include, red for reject, yellow for come back later. The magic is in the speed. 100 abstracts in under an hour if you trust your instincts. Don't overthink it. Separate the obvious winners from the obvious losers. The maybes get one more look at the end. Here's the speed principle. 90% of your screening decisions come from the abstract alone. You don't need the full paper. You don't need the PDF. The abstract tells you what they studied, how they studied it, what they found. That's enough to know if it belongs in your review. Stop reading entire papers just to decide if they're relevant. That's backwards. Read the abstract, make the call, move on. Full text is only for the final 15, the papers you're actually synthesizing. Once you've got your 15 anchor papers, now the real work begins, and it's actually fun. Finman extracts what I call the DNA of each paper. Model, results, gaps, all pulled into one synthesis matrix. You see at a glance how these 15 papers compare. Where do they agree? Where do they conflict? What gaps do they all mention but nobody fills? This matrix is gold. It's not organizing, it's thinking. Your paper starts writing itself because you see the patterns. The structure emerges from the data. That's when research gets exciting. Look at this, a transparent trail. Records identified, duplicates removed, records screened, final 15 included. This isn't just for you. This is your receipt. When a reviewer asks how you selected your papers, you show them this flow, permanent proof that your screening wasn't random or biased. Academic rigor isn't about reading more papers. It's about defending your choices. Every decision logged, every exclusion justified. You're not just doing a literature review. You're doing one nobody can question. Here's the payoff. Once you hit those 15 anchor papers with their DNA extracted, your paper outlines itself. Themes emerge, gaps become obvious. The structure you've been stressing about just appears. People spend months stuck on, how do I organize this? Then they build a synthesis matrix and the answer is right there. Your job shifts from searching to synthesizing, from collecting to creating. That's the transition every researcher wants. The 15-paper method gets you there without the burnout. I'm leaving you with this. Stop reading. Start screening. Reading every paper is a trap. Feels productive, but it's not. Screening is productive. Filtering is productive. Finding the 15 papers that actually matter and ignoring the 9,985 that don't, that's how you finish without losing your mind. The tools exist. The method is proven. You don't have to suffer through research anymore. Your next paper is waiting.